Dear students, in this lecture presentation, we will learn about the insulin actions and we will have an introduction to the insulin preparations. This photograph shows Banting and Best, two people who were associated with the discovery of insulin in the year 1921. Also seen is one of the experimental dogs which was used for the discovery of insulin. This photograph shows the first diabetic to receive insulin on January 11th, 1922. His name was Leonard Thompson. He was a type 1 diabetic. He received the injection of insulin in Toronto Hospital, Canada. So this is the insulin injection. Insulin cannot be given orally because it is a peptide. It gets degraded in the GIT. Insulin is administered subcutaneously. Insulin syringe on the left side and insulin pen on the right side are the devices which are used to inject insulin. The actions of insulin, basically insulin is going to enhance the disposal of meal derived nutrients, glucose, fatty acids and amino acids. Insulin also favors the storage of fuel in the form of glycogen in the liver and the skeletal muscle. Insulin also enhances the synthesis of proteins and lipids. Therefore, insulin is termed as an anabolic hormone. Insulin's effects on carbohydrate metabolism. Insulin enhances uptake of glucose in the skeletal muscle and adipose tissue. Insulin enhances the utilization of glucose. Insulin enhances glycogen synthesis. Insulin interferes with glycogenolysis. Insulin inhibits gluconeogenesis. Insulin enhances glucose transport across the cell membrane. How? This is possible because insulin translocates the vesicles which contain glucose transporter 4 from the cytoplasm to the cell membrane. GLUT4 is a glycoprotein which enhances the uptake of glucose from the blood vessels into the cells. There is an exception here. Some tissues like the brain, the liver, renal medullary cells, RBCs and WBCs do not depend on insulin for the uptake of glucose. Another interesting thing here is exercise has an insulin sparing effect on the uptake of glucose. That means in the presence of physical activity, skeletal muscles can take up glucose independent of insulin. That is why in diabetics we always ask the patients to indulge in physical activity so that they can enhance the uptake of glucose without the interference of insulin. In this way, we can reduce the amount of insulin given to the diabetics. So the summary of this slide, in diabetics, glucose doesn't get transported across the cell membranes. It remains within the blood, giving rise to hyperglycemia. Insulin enhances glucose utilization through the formation of hexokinase or glucokinase, an enzyme which is responsible for phosphorylation of glucose into glucose 6-phosphate, so that there is going to be utilization of glucose to the formation of the fuel or energy. So what we understand here is in diabetics, glucose is not utilized by the tissues. Insulin enhances glycogenesis. That means it increases the conversion of glucose to glycogen by enhancing the synthesis of an enzyme called glycogen synthetase. So in the absence of insulin, there is reduced formation of glycogen. All the glucose stays in the blood itself, giving rise to hyperglycemia. Second thing is insulin interferes with glycogenolysis. That means in the absence of insulin, there is increased breakdown of glycogen into glucose. So in diabetics, what happens is there is excessive formation of glucose from glycogen because of increased glycogenolysis. That means the hepatic output of glucose is increased in diabetics because of glycogenolysis. Insulin interferes with gluconeogenesis. That means formation of glucose from non-carbohydrate sources. Non-carbohydrate sources can be amino acids, glycerol, and free fatty acids. So insulin interferes with gluconeogenesis because it inhibits the enzyme phosphoenol pyruvate carboxykinase. So in the absence of insulin, what is going to happen is there is going to be increased formation of glucose from the non-carbohydrate sources. And the summary is in diabetics, the hepatic output of glucose is increased not only because of glycogenolysis but also to a major extent through gluconeogenesis. So summary, in carbohydrate metabolism, as far as diabetics are concerned, there is excessive production of glucose. That is, hepatic output of glucose is increased because of glycogenolysis and gluconeogenesis. There is reduced transport and utilization of glucose, and there is reduced formation of the storage form of glucose, that is glycogen. So all of these result in hyperglycemia. There is reduced utilization of glucose, there is excessive production of glucose. Effects of insulin on protein metabolism. Insulin enhances protein synthesis. Insulin interferes with proteolysis. Insulin interferes with the breakdown of muscle proteins. So in the absence of insulin's action, the muscle proteins are broken down into amino acids. These amino acids are funneled into the liver, where they are converted into glucose, pyruvate, and urea. The urea is excreted, giving rise to a negative nitrogen balance. So in diabetics, there is gluconeogenesis from the source of amino acids. That means hepatic output of glucose is increased in diabetics. Now insulin also enhances the formation of proteins by making available the amino acids to the muscle tissues, and in the absence of insulin this doesn't happen, so muscle protein formation is reduced. That is why in diabetics the skeletal muscle mass is reduced. Effect of insulin on lipid metabolism. Insulin interferes with lipolysis. Insulin interferes with the formation of ketone bodies. Insulin interferes with lipolysis. So in its absence, the lipids are broken down into glycerol and free fatty acids. And these two, that is free fatty acids and glycerol, are converted into acetyl coenzyme A, which gets converted to ketone bodies. Ketone bodies can interfere with glucose utilization by the brain, and when excessive ketone bodies are formed, it can give rise to diabetic ketoacidotic coma. And free fatty acids with glycerol, they form substrates for the formation of glucose, that is gluconeogenesis. So the summary is, in diabetics, because of a faulty lipid metabolism, the hepatic output of glucose is increased, giving rise to hyperglycemia. So the actions of insulin can be rapid, intermediate and late. What all we saw now, the metabolic effects of insulin, they are all rapid. They take place within seconds to minutes. Whereas the effects of insulin in increasing the expression of glucose transporters and in the expression of enzymes which affect amino acid metabolism, they have a latency of hours, and we classify them as intermediate actions. Whereas the effects of insulin on the proliferation and differentiation of cells are called as the late actions because they take a lot of time. Now let us look at the molecular mechanism of action of insulin, that is the action of insulin on its receptors. Now insulin receptors are present almost on every cell in the human body. The density is the highest in the liver and the adipose tissue. Now based on transducer mechanisms, insulin receptors belong to the third family, that is enzyme linked receptors. The enzyme which is linked to insulin receptor is tyrosine protein kinase. So the insulin receptor, as you can see, is present on the cell membrane. It is a heterotetrameric receptor. It consists of two extracellular alpha subunits which have the insulin binding sites. It also consists of two transmembrane beta subunits which have the tyrosine kinase activity. Binding of insulin molecules to the insulin receptor gives rise to aggregation and internalization of the receptor. This causes activation of the tyrosine kinase, and activation of tyrosine kinase is responsible for phosphorylation of the tyrosine residues. So beta subunits, they are responsible for phosphorylation of the tyrosine residues on each other, and this is responsible for exposure of the catalytic binding site, and this is the reason for phosphorylation of insulin receptor substrate proteins or IRS proteins. And this phosphorylation of the IRS is responsible for a cascade of phosphorylation and dephosphorylation reactions in the cell, which is responsible for activation and inhibition of the various enzymes which are responsible for the metabolic reactions, which we have classified as the rapid actions of insulin. Insulin is also responsible for translocation of GLUT glycoproteins from the cytoplasm to the cell membrane, and this happens through the mediation of several molecules which we call them as second messengers. Then insulin is also responsible for regulation of the expression of various carriers and enzymes, and finally insulin is responsible for cellular proliferation and differentiation as a late action of insulin. Now let us look at the insulin preparations. Based on the source, insulin preparations can be conventional, highly purified, human insulin preparations, and insulin analogues. The conventional commercial preparations of insulin were derived from animals, that is the bovine and the pork pancreas. The impurities were very high, up to 1% or 10,000 parts per million, in the form of proinsulin, insulin derivatives, pancreatic proteins, and other polypeptides. This was the reason for the high incidence of anaphylactic or allergic or hypersensitivity reactions with the conventional preparations, and because of so many antigens, antibodies were produced which were responsible for insulin resistance, because these antibodies get bound to the insulin molecules and inactivate them. Conventional preparations were also responsible for injection site lipodystrophic reactions, because of which they are not preferred. Except for economical considerations, we do not use conventional preparations. Because of high amounts of impurities in the animal derived insulins, purification techniques were developed in the form of gel filtration and ion exchange chromatography, because of which the impurities were reduced to less than 10 parts per million, giving rise to highly purified preparations which were called as single peak and monocomponent preparations. Monocomponent porcine insulin is as immunogenic as human insulin. That is, it is highly stable and the incidence of allergic reactions is minimal or even absent. Number 3, based on the source, we have human insulins. So human insulins are derived from recombinant DNA technology. Escherichia coli cells are very commonly used. Now let us see what is this recombinant DNA technology. In recombinant DNA technology, we remove the plasmid from the E. coli cells. Plasmid is an extra chromosomal DNA. The plasmid is cleaved or broken open with the help of a special enzyme. Now what we do is we introduce the DNA which codes for the human insulin into this broken plasmid. Next, this is recombined. The plasmid is closed with the help of another special enzyme. The recombined plasmid is now introduced into the host E. coli cell, and this host E. coli cell starts replicating into many daughter cells, and all these daughter cells will have the gene which codes for the human insulin, as a result of which all these cells starts producing human insulin, which is collected and used for the treatment of human diabetics. The advantages of human insulin, which is produced or synthesized by the recombinant DNA technology, is that it is similar to the endogenous insulin. There is no immunogenicity because there are no impurities and there is no injection site lipodystrophy. It has a slightly more rapid onset of action, a quicker and a consistent peak action, and a slightly shorter duration of action. Number 4, insulin analogues. These analogues are pharmacodynamically similar to human insulin but kinetically different. They are also manufactured by the recombinant DNA technology, so they have the same advantages as that of human insulin. So insulin analogues can be rapidly acting or they can be long acting peakless preparations. Insulin lispro, insulin aspart, insulin gluconeogenesis are the rapidly acting insulin analogues. Insulin glargine, insulin detamir, insulin digludec are the long acting peakless insulin analogues. Till now we saw the classification of insulin preparations based on the source. Now we shall see the insulin preparations based on their pharmacokinetics, that is the onset of action, the peak action and the duration of action. So once again, under the rapidly acting insulin preparations, we have the 3 rapidly acting insulin analogues: lispro, aspart, gluconeogenesis. Short acting insulin preparation, we have regular or soluble or plain insulin. Intermediate acting preparations, lente insulin, isophane or neutral protamine hagedon, NPH insulin. Long acting insulin preparations are the long acting and peakless insulin analogues, glargine, detamir, degludec. So this is the classification of insulin preparations based on their pharmacokinetics. So this is a picture from our textbook KD Tripathi. So you can see there the rapidly acting insulin preparations, the onset of action is 0.2 to 0.3 hours. That means 12 to 24 minutes. Short acting insulin, regular insulin, has an onset of action 30 to 60 minutes, 0.5 to 1 hour. Intermediate acting preparations, lente and isophane insulin, the onset of action is 1 to 2 hours. Long acting preparations, glargine and detamir, the onset is 2 to 4 hours. Have a look at the peak action. Rapidly acting preparations, they peak after 1 to 1.5 hours after injection. Regular insulin peaks at 2 to 3 hours. Intermediate acting preparations 8 to 10 hours, and the long acting insulin analogues, as I've told you, they are peakless, they do not have a peak. The duration, rapidly acting preparation, they act for 3 to 5 hours, regular insulin 6 to 8 hours, intermediate acting insulins 20 to 24 hours, and the long acting preparations, they act for 24 hours. Thank you very much. I will follow this with the next presentation where I am going to explain you all these preparations in detail. So thank you very much. Happy reading.